Hello, my name is Makala Faniel, and today I will be presenting my research entitled Kinetic Analysis of Bispecific Molecules Linking T-cell Receptor and Tumor Antigen for Cancer Immunotherapy. T lymphocytes, or T cells, are critical to our immune system by identifying and destroying infected or cancerous cells. They are activated when the T cell receptor, or TCR, engages with an antigen peptide presented by the major histocompatibility complex, which triggers T cells to produce cytokines and kill target cells. Cancer cells are often able to avoid T cell detection due to multiple mechanisms, including low reactivity to TCRs, which allow them to undergo uncontrolled growth. Bispecific molecules are an attractive concept for cancer immunotherapy due to their ability to simultaneously bind to multiple targets. This project analyzes a set of bispecific molecules developed by UK-based biotech company Immunocore. These bispecific molecules, which I will call MTACs, consist of an engineered TCR that binds to cancerous PHLA and an anti-CD3 single-chain variable fragment that binds to the CD3 protein complex of T cells. My project focuses on the analysis of these developed MTACs for future implementation as cancer therapeutics. AIM-1 seeks to identify the determining factor of MTAC anti-tumor efficacy. This will be achieved by measuring the 2D binding affinity and force-dependent bond lifetime of the MTAC molecules for cancer PHLA and CD3. These biophysical measurements will then be correlated with functional assays performed on MTACs. In my second aim, I will assess how TCR docking orientation affects anti-tumor efficacy. Similarly to AIM-1, I will be measuring affinity and bond lifetime, specifically looking at TCRs in canonical or reverse orientation. Additionally, I hope to determine if the absence of CD8 is responsible for low efficacy of reverse orientation TCRs. My project will utilize three main assays. The adhesion frequency assay is used to measure binding probability as a function of contact time. The system uses red blood cells coated with soluble proteins of interest, and through this study, the binding affinity and off rate of ligand receptor pairs can be derived. The biomembrane force probe is a JULAB specialty which measures the lifetime of a single bond as a function of tensile force. Through this technique, I am able to observe the formation of catch bonds, in which lifetime increases with force, or slip bonds, where lifetime decreases with increasing force. Lastly, the calcium flux assay visualizes MTAC ability to stimulate T cell activation by imaging the release of fluorescently labeled calcium when in the presence of MTAC molecules, which can be quantified to determine differences in the overall strength of activation. I have measured adhesion frequency relationships for S1 and S2 TCRs against PHLA, with S1 showing an affinity 1 order of magnitude higher than S2. Additionally, I have measured the U0 anti-CD3 affinity for CD3 to be consistent regardless of the TCR present. As expected, bond lifetime profiles for S1 and S2 TCRs against PHLA show catch bond formation. The calcium flux assay showed all MTAC configurations were able to activate T cells except for the S4U0 model, which was expected as S4 is our only reverse orientation TCR. In the coming months, I hope to expand upon this work by measuring the S4 TCR PHLA binding affinity and bond lifetime relationship. I expect the S4 TCR to have a lower affinity than S1 and S2 and to form a slip bond. Additionally, I will be measuring the binding affinity of the second anti-CD3 complex, U28, which is expected to have a lower affinity than U0. I would like to acknowledge my institution, PI, collaborators, and funding support.